Welcome to our deep dive. Today we're looking at a really fundamental shift in how we understand our own bodies.
That's right. We're talking about the 2025 Nobel Prize in Physiology or Medicine.
Awarded to Mary Brunkow, Fred Ramsdell, and Shimon Sakaguchi. And it's all about this crucial idea of immune restraint.
Mm-hmm. Yeah. And our goal here is to really unpack that, to get into how the body stops its own defenses, its immune system, from attacking itself. We call it peripheral immune tolerance.
And this isn't just theory, right? This has huge implications.
Oh, absolutely. It's directly relevant to how we're thinking about treating things like cancer, but also autoimmune diseases. It cuts both ways.
Okay, so let's dig into that. The immune system's big job is telling self from other, invaders. What happens when that goes wrong? That's autoimmunity, isn't it?
Exactly. That's when the system attacks the body's own tissues. And for a long time, the main explanation focused on something called central tolerance.
That happens in the thymus, right? Like
Yeah.
a training ground.
Precisely. Think of it like immune system boot camp. T cells that react too strongly against self are, well, supposed to be eliminated there.
Supposed to be.
Right. It's a neat idea, but it turns out it's not foolproof. It's leaky. Some self-reactive cells get out. And that's where Shimon Sakaguchi's work in 1995 really changed the game.
Ah, so he found what happens after boot camp, out in the field, so to speak.
You got it. He discovered this whole other class of cells that nobody really understood before, regulatory T cells, or TREGs.
TREGs, okay.
And these cells weren't fighters. Their job was different. They actively calm down immune reactions out in the body, in the periphery. They prevent the system from overreacting or attacking itself.
So they're like the immune system's own internal police force, keeping the peace.
That's a great way to put it. Yeah, the security detail. But, knowing the guards exist is one thing. How do they get their orders? What's the switch?
And that's where the other two laureates, Brunkow and Ramsdell, come in. This gets really interesting at the molecular level, I imagine.
It does. In 2001, they provided that crucial molecular piece. They were studying these mice, scurfy mice, they were called.
Scurfy mice.
Yeah, and these mice had this absolute devastating autoimmunity, multi-organ, incredibly severe, that their immune systems just went haywire, attacking everything. They died within weeks.
Wow, so a total system failure.
Catastrophic failure.
Yeah.
And Brunkow and Ramsdell linked it to a specific faulty gene, FOXP3.
FOXP3.
They also found that mutations in the human version of FOXP3 cause a really serious autoimmune condition in humans called IPEX Syndrome. It mostly affects boys because the gene is on the X chromosome.
So losing this one gene, this FOXP3, leads to immune collapse.
Essentially, yes, it removes the brakes. And then Sakaguchi put it all together just 2 years later.
He connected his TREGs to their FOXP3 gene.
Exactly. He showed that FOXP3 is the master regulator, controls the development, the function, everything about these regulatory T cells. No FOXP3, no functional TREGs, no self-tolerance.
So FOXP3 is the molecular switch that makes those security guards, the TREGs, actually work.
It's the key. This discovery completely reshaped immunology. We went from seeing the immune system as just, on or off, attack or don't attack.
To something much more nuanced.
Exactly. To a dynamic balance, a constant negotiation between activation and restraint. This whole field of peripheral tolerance research basically exploded from there.
And that brings us to today's applications.
Yeah.
Because this understanding lets us try to, tweak that balance, doesn't it?
Precisely. And it goes in two directions, which is fascinating. For autoimmune diseases, or say after an organ transplant, the goal is often to boost TREGs.
To strengthen the calm down signal.
Right. There are experimental treatments trying to expand TREG populations or make them more stable to reduce harmful immune attacks without just globally suppressing the entire immune system.
Like trying to improve graft acceptance in transplants by infusing engineered TREGs.
Exactly. But then you flip it for cancer.
Because in cancer, the TREGs are actually a problem.
They are often a major barrier. Tumors can be full of TREGs, and these TREGs suppress the immune cells that should be attacking the cancer. They're like guards protecting the enemy.
So for cancer, researchers want to do the opposite. Get rid of the TREGs, or at least stop them working near the tumor.
That's the challenge. How do you selectively dampen or remove the tumor-associated TREGs without removing the ones protecting the rest of the body from autoimmunity?
So it's not just an on-off switch we need, it's more like a dimmer, or maybe multiple dimmers in different locations.
You've nailed it. It's about selectivity, finding ways to reprogram or deplete only the TREGs inside the tumor environment. That balance, that's the frontier right now.
It's incredible science getting us closer to potentially curative approaches. But, the sources we looked at also raise a really critical point about access.
The cost factor?
Yeah. These advanced therapies, like engineering a patient's own T cells, they're groundbreaking, but they are also incredibly expensive right now. It's creating real gaps in who can actually benefit.
It's a serious challenge.
Which leads to a final thought for you, our listener. As we gain this amazing power to fine-tune our immune systems, potentially even control life-changing genetic mechanisms, how do we make sure the policies and, frankly, the ethics keep pace? How do we ensure these breakthroughs benefit everyone, not just those who can afford the high price tag? Something to think about.